But anyway, let's move on to the main topic of the day. And I am delighted to welcome Professor Ard Louis, a long-time friend of the Faraday. And Ard is going to be speaking to us on the topic of evolution, algorithms, and convergence. And if I can just do a brief introduction for us. Ard is Professor of Theoretical Physics at the University of Oxford, where he leads an interdisciplinary research group studying problems on the boundary between chemistry, physics, and biology. He's also Director of Graduate Studies in Theoretical Physics. And before his post at Oxford, he taught theoretical chemistry at the University of Cambridge here, where he was also Director of Studies in Natural Sciences at Hughes Hall. From 2002 to 2010, he was a Royal Society University Research Fellow, also previously a member of the Biologos Board of Directors, member of the ISSR and the Faraday Institute International Advisory Board. He's also turned his hand to co-presenting the 4-part documentary Why We Are Here with David Malone, and appeared in The Story of God with Morgan Freeman, our TV production. Ard was born in the Netherlands, raised in Gabon, and received his first degree from the University of Utrecht and his PhD in theoretical physics from Cornell University. So, as I say, we're absolutely delighted to have you with us, and I will hand over to you. Thank you very much.
Yeah. It's a pleasure to be here at the Sicily Friends in the audience. I'm Howie. And I want to tell you about some ideas I've been thinking about a lot for the last. So these are ideas I've been thinking about for a while, and I want to illustrate them with this famous little short story by Borges called The Library of Babel. So this is a library where every book of 410 pages, 40 lines per page, 80 characters per line, with a 25-character alphabet. So every possible book of those characters was put there. So there's 1.3 million characters per book. So there's 25 to the 1.3 million, which is about 10 to the 1.8 million possible books. It's a very large library. And in this story, people live in the library, and they look through the library for things that they find interesting. And at some point, the librarian deduced that the library was total, perfect, complete, and whole. That is, all that is able to be expressed in every language, all the detailed history of the future, the autobiographies of the archangels, the faithful catalogue of the library, thousands and thousands of false catalogues, the proof of the falsity of those false catalogues, a proof of the falsity of the true catalogue, the Gnostic Gospel of Basilides, the commentary upon that Gospel, the commentary on that Gospel, the true story of your death, the translation of every book into every language, the interpolations of every book into all books, the treatise Bede could have written, but did not, on the mythology of the Saxon people, the lost book of Tacitus, etc. So part of the story is that people go through the library looking for their vindication, the story that's going to tell them the meaning of their life. But of course, as Borges points out, not only is that book there, but so is every possible treacherous variation of it. And so this is the stories. Every possibility is there. And so the interesting thing is possibility spaces are very, very large. A 400-page book is not that large, but every possible version of it is an extremely unimaginably large space. Every book is equally likely, okay? But is every story equally likely? You can think about this and pick up some early intuition of what I'm going to tell you, which is imagine stories that are relatively simple to tell. They can be told in many different ways. So there might be many books that are effectively telling you the same story. While a very complicated book, like the exact detail of what happened in your life, might need up to now, might need all 410 pages to be properly written down, and it'll only appear once or a very small number of times. This really gives you an intuition, which is that simpler stories should appear more frequently than more complicated stories. That's the first intuition. Just keep that in your head. I'm going to come back to it a few times. What is this possibility space of all possible stories like? And can we think of other possibility spaces? Well, biological possibility spaces are also unimaginably large. In fact, people often talk about hyper-astronomically large because when you start thinking about them, you're automatically drawn to metaphors larger than the universe of some kind. So proteins, very important molecules in our bodies, have an alphabet of 20 letters. So a protein of length L has 20 to the power L possible different sequences. How big is that? Well, I took every length 37 protein, it weighed more than the Earth, and made every possible length 58
peptide, really, protein, is way more than the visible universe. The mean length of proteins in your body is 476. If I made every length 476 protein, it'd weigh 10 to the 500 times the weight of the observable universe, which, if you're into numerology, is what my string theory colleagues tell me is roughly the number of minima in the Calabi-Yau manifold, so the number of possible multiverses. Clearly there's a link there of some kind. I'm going to look not so much at proteins, but at RNA. So you may remember DNA, RNA, and proteins are the key elements in of the kind of genetic makeup of your body. And RNA is quite special because it can both encode information, but it can also be active. Like proteins, it can be a catalyst, or it can be structural, so it has functional behavior. And a lot of scientists, including myself, think that a very likely possibility for the origin of life, how do you get both catalysis and information at the same time? Proteins do catalysis, DNA is information, RNA can do both. There may have been an early version of life that was based only on RNA. RNA has a 4-letter alphabet, so doesn't go quite as fast, but still true that, every length 55 RNA would weigh 10 to the 10 kilos. Only 79 would weigh more than the Earth. And if you made every length 126, you're still a very short RNA, actually. If you made every possible one, it would weigh more than the mass of the observable universe. So that's a very big space, like that space of Borges's library. You might ask yourself, what can I find in that space? Are all possible biological things there? Is that world full of function, or is it full of nonsense? What is that space like? And this idea, you can find it throughout the history of modern biology, particularly when people start thinking about sequences, DNA sequences. They quickly realized that these spaces were unbelievably large. And so the kinds of picture, if this is a picture with two axes of information space, then one possibility may be that space is hyper-astronomically unimaginably large, and life as we know it started somewhere fortunate and hasn't really had time to explore very much of that space because it can't, because the space is too big to explore. And so this is the only part of that whole space that's actually functional. Or it may be that there are big parts of that space that are functional, like these white islands here, and they maybe were in this one, and there's another whole different kind of life that could have emerged that is like ours, but different, or maybe very different, right? That possibility. That possibility is actually there's many, many pockets of life, and maybe they're all different, maybe they're all similar to each other, and you could probably think of other ways of thinking about this. But clearly, those kind of questions arise, like how lucky are we to actually have life on our planet? If you were to have, start biomolecules somewhere else, and they didn't start in quite the same space, would life emerge again, or would they just be unlucky? Those are the kinds of questions that you might ask. And if you look back in history, a lot of people have also worried about that. So I'll call this the Salisbury-Hoyle paradox. I have the two famous scientists who thought about this. So Frank Salisbury wrote a famous paper in Nature in 1969 about hyper-astronomical biological spaces, and basically pointed out these numbers of this size have no precedent. And so the famous Sir Fred Hoyle, also here in Cambridge, wrote close to the end, actually not that close to the end of his life, but in 1981, when he was starting to speculate about biology, I was constantly plagued by the thought that the number of ways in which even a single enzyme, that's a protein, could be wrongly constructed was greater than the number of all the atoms in the universe. So these kinds of ideas there's worries that, if the space is so big, how are you going to find function in it, and how isn't it going to be very easy to break function? And there's a very famous conference that happened in the 1960s called the Wistar Conference, where some mathematicians and biologists, a lot of famous biologists, chaired by Sir Peter Medawar, Nobel Prize winner from Oxford, and Murray Eden, a very famous engineer, mathematical engineer, said, either functionally useful proteins are very common in this space, so that almost any polypeptide one is likely to find has a useful function to perform, or else the topology appropriate to this protein space is an important feature of exploration. That is, there exists certain strong regularities for finding useful paths through this space. And this is a theme you'll find repeatedly when more mathematically oriented scientists look at this space. They say, well, these spaces are too big to find something. There's a problem here. And actually, this Wistar conference is kind of fun to read because it was a discussion conference, and the person who transcribed it didn't edit it at all. So you see some fairly strong language being used and some strong opinions being expressed, unfiltered. And basically, the response to Eden by Richard Leontin, another famous evolutionary biologist, was, look, here's proteins, they work. I see that you think there's a problem, but there isn't a problem because we see them empirically all the time. And that's the kind of answer that was often given to people like that. And this is why you saw Salisbury saying, I documented in a paper in Nature magazine, this is about maybe 30 years after he wrote this, before he died, actually maybe 40 years later, my arguments have never been refuted, just ignored. So the feeling by some of these people are that these arguments are being ignored. And this brings me to an interesting link to the science religion discussion. There's another local Cambridge link, in fact. This is Doug Axe, who in the early 2000s was working here at the LMB and wrote a paper where he tried to estimate how likely it was to find a functional enzyme, so a protein that does catalysis. In this particular case, for the particular protein he looked at, he said it's a probability of 1 in 10 to the 77. And these are effectively, again, unimaginably large spaces, unimaginably unlikely to find. And so this argument that basically you couldn't find function in this very large space unless you did something very special has been, I think, it's probably the most important argument in the intelligent design discoveries style rules. If you start pushing on a lot of the rhetoric, and it's a lot of rhetoric that they use, this is a key argument that underlies it. Basically, the space is too big and therefore you can't find function. Therefore, something outside of natural processes would be needed to be able to explain how you find function. And so if you think these spaces are very big, they're unsearchably big, at best, we got lucky, okay, and we got to a space where there is some function and we can kind of look around there and find some local function, and maybe there's something very special about where we started, but the space is so big, it's unlikely you're going to find the same thing twice. And this, I think this idea plays in the background to a very common sense among many of my biological colleagues that, life is radically contingent. In other words, if you were to replay the table of life again, you get something completely different. Space is unimaginably big, you'll end up somewhere else, it's going to be different. You have no idea where it's going to be, and so it's going to be radically, absolutely different. The chance of the same thing repeating itself is incredibly small. Now, here's a very different story I'm going to tell you, which is a story that Simon Conway Morris introduced me to. So I've got 2 of his books here, and you'll see, Simon, in a minute why this was very important in my own development. So these are, as you've probably seen if you've come to lectures at Faraday or read a bit more, there's a lot of evolutionary patterns that repeat themselves again and again and again. And they go all the way down from the microscopic molecular level all the way up to the way our brains work. And in these books, Simon showed this book, Life Solution, and more recently, The Ruins of Evolution, a wide number of examples. So I'm just going to give you one example, which is one of my favorites. These are anteaters. So each of these mammals is very odd and strange. They have these bulging salivary glands because they eat ants. They got ants don't like to be eaten. They've got a long kind of hot dog-like snout. They've got almost no teeth and a stomach as rugged as a cement mixer because ants don't like to be eaten. And so you might think, well, that's obviously very well adapted to this particular niche, but you would expect something that strange and that odd to really only evolve once. But here, look, there's a bunch of them around different parts of the world. There's a giant pangolin in Africa. In fact, I grew up with pangolins around in the jungle where I lived. I often saw them. Giant anteater in South America, the armadillo in North America, and the spiny ante in Oceania. Each of these animals actually evolves completely independently of each other. In fact, the spiny anteater lays eggs, no relation at all to the other animals. So although this space is very, very large, okay, somehow the same solution, in this case, eating ants, was found multiple times. Now, one argument would be, shows you the power of natural selection. Natural selection is very powerful. There are certain niches, and what you find, surprisingly, is that forms will evolve into the shape of that niche. And so that's why you see multiple anteaters evolving. As still, I think, leaves up some open questions. So we clearly have observed this. This is true. These animals are able to evolve into a very specialized niche relatively rapidly and independently of each other. And that does tell us something about the power of natural selection. But I think there's something more going on in the background, perhaps, than just that. Because it reminds remarkable and surprising. And I think when you read Simon's books or other people that wrote on convergence, the most common kind of adjective is just surprise and bafflement. Like, how could this happen? I think for a scientist, like physicist like me, not a scientist, a physicist, this is what got me interested in evolution, because before that, if it was just contingent, if it was just one damn thing after another, then there's not a lot for a physicist to study, okay? It's historical. You have to go back in history. It's depending on exactly what happens. That's fine if that's the kind of work you want to do, but physicists like patterns that repeat, right? And so I didn't, as many of my colleagues said, that we aren't interested in evolution. And I spoke with many of my physics colleagues who tell me that reading Simon's books were what got them interested in biology and in evolution. Not long before I left Cambridge to go to Oxford, I met with Simon. He said to me, you're a physicist. Couldn't you find an equation for convergence? And I left thinking, I don't know. I had no idea how to even ask that question or formulate that question. But I was thinking about it. And one of the examples that Simon pointed out to me was one that I thought maybe I could have a stab at. And that's this experiment by Jack Szostak, who's a Nobel Prize winner, very famous also for work on the origin of life. And what you can do for molecular evolution is do this in a test tube repeatedly, and therefore you can control a lot better than you can for anteasers what happens. And so what he did is he took a bunch of random RNA strands of length about 55. Remember, 55, there's 10 to 10 kilos if you make every possible strand. So he only used, I estimate, about a hundredth of a gram of material, so about 1 in 10 to 15th of the space. And he had an assay which was basically looking for a certain type of catalytic activity called self-cleaving enzymatic activity. And you run your experiment and you enrich your on your assay so that you get closer and closer to something that does what you want. And what he showed is they were able to find this kind of function. But really interestingly, every time they did the experiment, they kept finding the same shape. It's called the hammerhead ribosome. Not so why it's called the hammerhead ribosome, but it's a particular way that this thing folds. So this strand folds into a certain three-dimensional structure. That structure is really important for its function, and it finds the same thing again and again. I thought, well, here I might have a chance of doing something with physical techniques, because it's a really simple experiment. It's also very remarkable results, right? Because it's almost like you put your hand into a haystack, and every time you put it in, you find a needle. That's the probability that this would happen if this was a completely random, uniformly random process, which is a hidden assumption in a lot of the impossibility arguments about the size of genotype space. It's so big, you'll never find the same thing twice. The assumption is it's uniformly random, right? So something is strange in this story. And the question is, what is the cause of this? How come he keeps finding the same thing again and again? He's not selecting, by the way, on the hammerhead ribosome shape. He's selecting on an activity, right? So why does he keep finding the same shape each time? Yep. There's many, many possible shapes. But how is it possible? Well, one reason why I got interested in it, Simon, is because there is a very efficient algorithms for calculating not quite the 3D structure, but something somewhat similar to the question, which is the 2D structure, which is called secondary structure, 2D secondary structure, which basically tells you who binds to whom. So this is a sequence, GAGU, basically to first order A's and U's bind and G's and C's binds. And so if I give you this strand and I ask you, what would be the lowest free energy state? So what would the state that minimizes the maximizes the entropy and maximizes the energy, the entropy at the same time? That would be a state that basically this would be an example of it. So here you see it starts with C, then the A that starts with the G, then the UU, right? There's a GA. These bind to each other. There's a bit that don't bind, and then there's a bit here they bind. Notice the hammer. You see the hammer? Right? That's what's called the hammerhead ribosome. It's a fascinating thing. It also evolves independently across every major branch of life. So it's seen all over through life. We think it's convergently evolved many times. And now Szostak was able to make it convergently evolve in his laboratory. So the question is, what's the mechanism by which that convergent evolution happened? Maybe we could try to solve this simpler problem. I think one thing to remember immediately is that it can't be natural selection that gets you there, because you're only searching an incredibly small fraction of the space. And just to remind you how that works, so in Darwinian evolution, there are two steps. There's variation and natural selection. So in modern we would say mutations cause variation. So your offspring are different from you. And if those offspring have some fitness advantage, so for example, maybe they have more viable offspring than you would, then that trait will end up fixing in a population. That's what's called natural selection. But the first step is also you need to actually have that trait first. That's called variation. So variation proposes and natural selection and disposes. That's the simplest way of looking at it. And in fact, that a very famous quote by my countryman Hugo de Vries, who rediscovered Mendelian genetics, which Darwin was unaware of or not very aware of, said natural selection may explain the survival of the fittest, but it cannot explain the arrival of the fittest. So in this hybrid ribosome question, because Strotek is only using 1 in 10 to the 15th of all sequences, there is an arrival of the fittest problem. He's only using an incredibly small fraction of all possible sequences. Natural selection can only work on that which is given. It's only given a tiny fraction of the set of all possibilities. So therefore, that must be part of the story. And the question is, how far can I push this part of the story, the variation, and what can I explain from that? So to do that, I want to take a step back and think a little bit about a bigger picture question, which is a question about algorithms. So what are algorithms? Well, algorithms are just names for the things that computers do when you calculate something. And I'm going to give you 2 intuitions for algorithms. One is a very, very dumb algorithm. It's monkeys on typewriters. Can monkeys on typewriters type? Will they ever reach right there? Works of Shakespeare in theory, yes. On practice, if they're typing on a typewriter, every input is equally likely as every output is equally likely as any other output of a certain length. If you have k letters in your alphabet, probability you get a length L string is 1 over k to the L. But there may be another algorithmic way of thinking about this, which is slightly more clever, which is a monkey typing into a computer program like Python or C. Right? Then there are certain types of outputs that if the monkey types a short code like print 01 50 times, you'll get a long output of 100 01s. So therefore, if I'm looking at the output of a monkey typing into a typewriter or a monkey typing into a computer, or for those who don't know what typewriters are, monkey typing into a word processor and into a computer language, I can tell the difference because certain strings with short description are going to appear exponentially more often in the second case than the first case. And that's important because when I think about Shostak experiments or any kind of evolutionary system, right, the monkey typing is like the random mutations, okay? Those are actually not, you are not your DNA, right? You are the process of a developmental program that takes that DNA and develops it into you. So really, you are the output of a certain random typing initially into a computer program language that generates certain outputs. And so that second image is the right way of thinking about mapping from sequences to phenotypes. So here's a little thing to think about. Another way of thinking about these kinds of questions is to ask a very different question. So I'm going to take a step to something which is going to try to be linked, but I'll link it back in a second. So this is a slightly busy slide, but just to remind you, for this, I think binary strings, if you're in computer science, you'll know that everything can be turned into binary. Remember, there are 4 strings of length 2, there are 8 lengths strings of length 3. So there are 2 to the n lengths strings of length n. So if I take this example here, if there are length 10 strings, it's 1,024 strings of length 10. And let's say I have a compressor, so something like you zip it. I want to have a perfect compressor, so I need it to describe my length 10 strings by strings that are shorter. How many of those strings do I have, right? So if I actually add up all of these strings that are shorted together, I only have 1,022 strings. So there's 2 strings that I can't compress because I don't have enough. I can only compress them with something that's shorter than them. Right? About half of those strings can be compressed by 1 bit, 3/4 by 2 bits, etc. So in general, if I take all possible outputs, only an incredibly small fraction of them can actually be compressed. When you go on your computer, you compress a file, like a book, into something much smaller. That tells you that that's an extremely rare sequence of symbols. Because if you compress it by, hundreds of bits, the probability you could do that is 1/2 to the power hundreds. It's an unimaginably small fraction. So this tells us that actually, although Borges' library is unimaginably large, only an unbelievably small fraction of those books actually have semantic information that's interesting for us, right? Because they're compressible. That's a little principle to think about. So Therefore, in that biological space, the vast majority of these sequences are also random in this way. Now, why is this interesting? There's a link here, actually another Cambridge link. So Alan Turing at King's College, when he was very young, he's still 23, came up with what's now considered to be the foundational model of computation. It's called the Universal Turing Machine. I have a little picture of it. It works with tapes. And you can think about that as something, a very simple way of understanding computation. Interestingly, Turing didn't come up with this model in order to found the theory of modern computation, although that's what he did. He actually did it to show that there are things that computers cannot calculate. It's linked to Gödel undecidability. You see that as well in the title, the Unscheidungsproblem, which is a famous problem by Hilbert. And for those of you mathematicians, you'll know what this is. So that's a fascinating little aside. But you can ask yourself, what happens if a monkey types into a universal Turing machine? How likely will it produce a certain output or other outputs? And one of the principles of this whole idea of algorithms is, yes, although it does depend on which universal Turing machine or which computer you use, how likely certain outputs are to happen, actually, you can do say a lot about the likelihood that certain things happen up to constant terms. You can always write a compiler from one language to the other. So many things are not actually that dependent on the details of the computational model that you use, as long as it has certain types of power. For example, if it's universal, I can say a lot about it without needing to know exactly what language you're writing in. And the reason I'm saying this is important is because it's going to come back in a minute. And if that's true, that I can therefore look at biological systems without necessarily knowing what the language is in detail, what the algorithms are. I can tell you something about general properties of how that system is going to work. That's an abstract theoretical claim, but if you're in computer science, this is completely standard. But people outside computer science often worry that, what's the language? Why is he going to talk about algorithms in biology when he doesn't know what the algorithms are? And the claim is, I can do that because these principles are bigger. And one of the principles I'm going to use is one that comes from this man, Ray Solomanoff, a very eccentric American computer scientist who basically asked himself the following question in the 1960s: what happens if I randomly type into a universal Turing machine? So this is the probability called the algorithmic probability that a universal Turing machine U produces an output X, like 0, 1, 0, 1, 0, 1, 0, 1, or pi, or whatever it is. You have some output you want it to produce. So that basically, how likely is that? So I'm going to get into binary programs because computer scientists like everything in binary. So how likely am I to get an output like 0, 1, 0, 1 1,000 times, whatever you would like? Well, it's going to be sum over all programs that generate that output. Okay. Since I'm randomly typing in, the probability that I get is a string of length L is 1/2 to the power L, right? Because I'm writing everything in binary, so it's 1/2 to the L. So if my code is 10 lines long, it's 1 in 1,024 that I get that one. If it's 11 long, it's twice as unlikely. And so he pointed out that the probability that I get that is the first term in that sum is the shortest program. And this is called growth complexity. It's a very profound mathematical object. And it's the shortest code that will generate any object. And intuitively, this tells me that objects that have short descriptions are much more likely to appear if I randomly type into a universal Turing machine than obviously have long descriptions. So the point is, my intuition, hand-waving about monkeys, has been completely mathematically formalized in this field of mathematics called algorithmic information theory. And it's an important one. So Marvin Minsky, one of the founders of modern AI, said not long before he died, it seems to me the most important discovery since Gödel was discovered by Chaitin, Solomanoff, and Kolmogorov, the concept called algorithmic probability. Everybody should learn all about that and spend the rest of their lives working on it. So Simon, when I was thinking about what equations should I do for convergence, I used up at least 2, now I'm my 4th PhD students, looking at the mathematics of this. So this mathematics linking to Turing and to Gödel and these people is linked to these ideas of hopefully evolutionary convergence. So what this tells you, as I said, most books in Berger's library, most books have no meaning, but also simple stories, so stories that have short descriptions, are exponentially more likely to appear than complex stories. And you can think of other ways of making that analogy work. And so what's fascinating is this principle of simpler things being more likely than complex ones is much more general. So I'm going to briefly talk about a completely different line of work that my group works on, which is neural networks. So whenever you're hearing about modern AI, ChatGPT, they're all using something called neural networks. And neural networks work a little bit like this. Okay, so you've got some input. Okay, the input could be an image of a cat or a dog. And then you've got a bunch of hidden layers that you don't see. Okay, and they've got lots of little weights, often millions and billions of weights. Okay, little values. And then there's output. So for example, if you want to teach this thing to recognize and distinguish cats and dog pictures from each other, you give it a whole bunch of pictures of cats and dogs, you see what it tells you, whether it tells you cat or dog, and then you adjust these weights, you train these weights until it gives you the right value. What this thing is really doing is it's taking an input and has some algorithm that recognizes cat or dog. Okay, it's a mathematical algorithm. Interestingly, we don't really know exactly what that algorithm is. This is why they're so hard to interpret. This is why there's all these problems about security with them. But we do know, I mean, we can write it down in terms of all the weights, right? I can write it down, but I don't know what it actually is doing. But I do know that it's actually sampling over algorithms. So I can turn this question around and say, what if I'm a monkey and I'm typing at random in those weights, okay? What algorithms is my neural network going to produce? Okay, so here I've got on the left side a probability versus complexity. This is actually a compression thing for different functions. And what we show in this paper is that neural networks generically, okay, are going to be exponentially more likely to produce simple outputs, functions, and complicated outputs. I'm not going to tell you what this thing is doing. It's reading Boolean functions. But the interesting thing is the simplest Boolean function, which basically says everything is true, everything is false, appears about 10% of the time if you just randomly type into this thing. So this particular algorithm is a very simple one. It appears with high probability. And this bound that we've derived here, this is a bound we derived from algorithmic information theory, gives an incredibly good upper bound to what we observe in this system. And the reason I want to point this out is because when I start talking about evolution and biology as algorithms, people worry that I'm trying to say that DNA is like a computer code, right? It is and isn't, right? Just like the weights here, because you think of the weights here like the genetic information of an organism, you type on it, it's like random mutating, and the function that the neural network produces is like the phenotype. And so that principle tells you, even though I don't necessarily know, in this case, I don't exactly know what the algorithms are, but I can predict something generic about them, that simple ones are more likely to appear than complex ones. So now I'm going to try that same on this map. Okay, so I've got sequence that maps to a structure. That map, in this case, is using some thermodynamic information, minimizing free energies. Okay, I can write that down in a number of lines. Okay, but I don't really know what the algorithms exactly are. I can guess at it. And so the question would be, if I randomly take sequences, how likely am I to get certain structures versus other structures? And my prediction is I'm going to find simple ones much higher probability than other ones. So a nice thing about secondary structures is I can abstract them out a little bit. So here's a little short secondary structure. It starts here, G, C, C. The green things means a bond, so it binds, doesn't bind, binds, and like that. So that's the sequence here. And then here I've just got a slightly simple description. So every time there's a bond, I open a bracket. If there's a no bond, I put a little dot. I close a bond, I close a bracket. Okay. And you can see that basically this and that are equivalent to each other. This is a bit easier to write down. Okay. And I can say, well, actually, if I look at this one, if I flip this G and the C around, I'm still the same bond. So there are many sequences that might have this as their lowest free energy structure. So then I can ask myself, well, if I just look at these, I know there's many more sequences than structures. Which structure are going to have the most sequences mapping to them? It turns out for length 55, which is the length that Shostak did the experiment, we can actually work that out. So the simplest structure, which is the one that doesn't bind at all, is the most likely to appear when you randomly sample sequences. I sample sequences at random and I say, how many am I going to get? I get this one with the highest probability. It's also got the lowest, it's the most compressible. So it's a very compressible shape because just dot 55 times. This one here is a bit more complicated, okay? This is on log 10, so it's 10 to the minus 7. It's basically 10 million times less likely this one to appear, okay? And it's got a much higher complexity. And here you see the log of the probability, and I basically have just zipped. This is literally zipping. Like I zip them like you do on your computer. And the structures that are easy to zip are high probability ones, and the ones that are hard to zip are low probability. And I want to show a small fraction of this. This space goes on for many more orders of magnitude, but I can't sample it, okay? But that hemorrhoid ribozyme is one of these high, relatively low complexity, high probability structures. So the argument is, what's happening in Sherlock's experiment is he keeps finding this same structure because it happens to be easy to find. In this particular case, I know that that sampling that happens there is searching probably in 1 in 10 to the minus 10 of the total space of structures. So there are many, many more structures. We think there's 10 to the 33 possible structures you can find for this length RNA. Okay? The vast majority of them are never found by nature because you would have to sample for a very long time over random structures that we're finding. So only a very small subset are found. I can make that argument even more general. So here I'm going to do it with my postdoc, Sam. Here it's a bit of a more complicated story, but I've got like 300 RNAs. It's much longer, of which there's an unbelievably large number of them. Okay, and if we randomly sample sequences, this is the distribution of complexities that we find, and these are the kind of structures that we look. If we randomly just sample structures, we can also look at all possible structures of like 300 and just pick them at random, then we find that they've got a much higher complexity than the ones that are random. So this difference between the ones that you find at random and the ones that you find when you search in the space of structures versus the ones you find when you look at random over the space of sequences is what I call developmental bias. This is the bias by development towards these simple ones that you find right here. It turns out these are actually on the order of, you have to sample 10 to the 30 sequences more than many more times the way to the Earth to actually find them at random if you looked in the space at random, right? But this really small subset is found by randomly sampling sequences. And here this is more for the biologists, and here I've looked at a wide range of different RNAs. So there's all these long coding RNAs, non-coding RNAs, tRNAs, SNO RNAs. All of these are ribozymes. They all do something slightly different. But what's interesting is if I look at the distribution of your complexity values, then I've got here a bound between plus or minus 4 standard deviations of what I get by random sampling. I only need to sample about 100, maybe 1 million, maybe 100,000 random sequences, and I find the whole distribution of complexity is found in nature. And I can do that. I can look at other things that are maybe easier to interpret. Like, here's a structure. Okay, these structures, they've got things like junctions. They've got loops and bulges. I'll look at junctions just here. Okay, this is the distribution of junctions. If you look at all possible implemented structures, this is the distribution of junctions that we find in nature. Okay, so it's shifted by a lot. Okay, and what's fascinating is this distribution of junction or distribution of loops or any kind of property you can think of these structures. The one that was showing is what you get if you just randomly pick sequences. Okay, and the natural ones have that. So that's kind of interesting. So the question would be, why does nature only use a small subset of them? Well, the answer is because these are the easy ones to find. These are the ones that are highly compressible. They're the ones that are simple, right? Those are the ones that nature finds. What's also interesting is that if you look at these structures and they're being used in nature as a ribozyme, as a catalyst, for example, and you mutate them, right? Then there is a purifying selection that will kill the ones that are mutated away and bring back the ones that are correct. So that makes you think, well, therefore, since there's natural selection on these structures, and we know structure and function are closely linked to each other, we might expect that these would have come about by some process that walks up some kind of fitness landscape. So they started out, not very fit, and then they're here. They're clearly at the peak of the fitness landscape. But what's interesting is we don't find any evidence at all for this. So the structure we find for RNAs are so random that we've not been able to find, looking at all the RNAs that we studied so far, any clear evidence for natural selection on a particular property, except possibly archaea RNAs are slightly more robust than you might expect from random sampling. But basically, these things look almost like they've just been randomly sampled. So what this is telling us is that for RNA, the space of possibilities is really a space like this, okay? Anywhere you start, you're going to end up with something very similar. And every single one of the little dots, which is a functional RNAs, all look like the other little dots, okay? Doesn't matter where you start. They're all a tiny, tiny subset of the whole set of possibilities, okay? Very small subset of them, and they all look like each other's. And so it turns out that if you want to find function in RNA, okay, it's apparently very easy. But once function is found, it then is preserved by natural selection. But as far as we can tell, we don't find any evidence. It may still be the case that natural selection has found it, but it's hard to distinguish it from just this random model. this tells us that it must be really easy to find function. And so I haven't answered this question that was posed by many people like Salisbury and Hoyle about how hard it is to find function in protein space. That's a more difficult question. For RNA, I think we've shown that it's super easy to find function. And this problem, I think, has been partially solved. All right, in the last few minutes, I'm going to give you just one or two more examples of how these principles work. And here's one for you that I think is quite fun. It's still new, still unpublished. So we got interested in the shapes of leaves. And the reason I'm putting this in is because I know whenever I talk to biologists, like real biologists, like the two characters sitting over there who work on real big things, their eyes glaze over a little bit when I talk about molecules. So I'm going to show you something that's closer to what Rob and Simon work on. So these are leaf shapes, okay? So you can go to large herbaria, like this herbarium in Leiden, and look at tons of leaf shapes. And you can then people have worked out really nice phylogenies. In fact, there's a very nice paper in Nature last year. The biggest phylogeny is of angiosperm flowering leaves. And there are techniques you can use by looking at these to rebuild phylogenetic trees, basically says who relates to whom. And one of the fascinating things is that by looking at these phylogenies, you can also work out the rates of transition between different types of leaves. So leaves are often categorized into different shapes. This is called unlobed, lobed, serrated. There's a few different shapes. And what we see in this plot here is basically a little way of showing you that leaves that look like this or like that, which are more complicated shapes, are much more likely to have transitioned towards a simple leaf than an unlobed leaf and the other way around. And in steady state, this gives you around 80%, 90% of leaves that are found are simple, and a much smaller fraction of them are not simple. So that's what we see just from phylogeny, so looking at evolutionary history. And we're not the first to have looked at this. We just looked at it we're the first to use this more recent phylogeny. So the question might be, why is that true? Okay, is that because nature likes simple leaves because they have some kind of adaptive fortune? And we know that actually leaves are directly taking light and turning it into energy, so they're super important for fitness. And I know there are for sure properties of like size of these are very heavily linked to the environment, but what about the shape? So what we did is the reason we picked leaves is because there's some very good models of leaf development. So these are gene regulatory networks plus a whole bunch of morphogens. If you're into leaves, it's pretty nice leaf development models that you can get all these different shapes from, okay? And so you can then ask yourself, what if I just randomly mutate this model, okay? What is going to happen? Am I going to get simpler leaves with high probability than complex leaves, or am I going to find them all with kind of equal probability? And it turns out that if you do this, you actually find that randomly mutating them gives you quite similar trends in the going from complex to simple leaves as found in the phylogenies. And so in this particular case, the argument is the same, although now it's for something larger and more something you can actually observe. Our claim is we can explain a historic evolutionary pattern of a preference towards unlobed leaf shapes purely from the fact that these are easier to evolve. And if you take a step back and just ignore all my mathematics and my waving my hands and my stuff about Turing machines and just think, why is it easier to make one of these leaves than one of these? Well, if you look in more detail what's happening, there's a bunch of genes that are regulating different parts of the growth of the leaf. And it just turns out that if you just look at those, if you break that whole complicated thing down in just a few key processes, it's always much easier to make a simple leaf than a complicated leaf. Complicated leaf, you get all the rates just right to get this complicated shape. But to get a simple shape, you can do it in lots of different ways. And if you actually look at that model and just maybe something very simple, ask what volume of its phase space gives you simple leaves, what volume of its phase space gives you complex leaves, it more or less recapitulates what you see in these evolutionary histories. Now, does that mean that natural selection wasn't at play in the history of these leaf shapes? I think it was, okay? But the convergence, as it were, repeated convergence and preference for this simpler leaf shape may be explained in this case by this principle that I'm telling you about. And this is I can explain it to you in a mathematically complicated way, or in this case, just on this model in a simpler way. So that's another example, and we're currently working on tooth evolution. In tooth evolution, you can also see without too much difficulty that random mutations are much more likely to go to simpler teeth than complex teeth. And there, as opposed to the two examples I just gave you of RNA and of leaves, we do see differences between randomly mutating and what you see in nature, not surprisingly, because teeth, again, are directly linked to adaptation. Right? But it is for sure true that simpler teeth are exponentially easier to evolve than complex ones. I've got now close to the end of my talk, so I've tried to look at these questions of deep structure biology. I actually just discovered this recently, that this is a book that I think I wrote in 2008, so around not too long after I had left. And I see that actually I say in the bottom of that, an immensely stimulating book. It draws you in with bold hypotheses and original perspectives. I was inspired to take a fresh look at my work and see if I could apply some of the ideas in my lab. So I actually, in this case, I wrote a blurb that I actually ends up believing and following. So thank you, Simon, for that stimulation. So what did I say today? I told you that biological possibility spaces are unimaginably vast. That, I think, is not controversial. I also am arguing that a better way of thinking about evolution does than searching randomly in the space of phenotypic possibilities is to say that it's searching in the space of algorithms. Okay? And that's compressible phenotypes, so ones that have short algorithm descriptions are exponentially more likely to appear when you look in that space. So although, for example, the set of all possible sequences of length 300 RNAs is unimaginably large, the set of phenotypes that you find if you pick half of those sequences is a super tiny subset of the total possibility space. So you're going to keep finding the same ones again and again and again. Even though that space is very big, you're only going to find a small subset of them. And those ones, I can tell you in advance, are going to be ones that are compressible. Okay, so these phenotypes are a tiny fraction of all possible phenotypes. So this is like an Occam's razor, right? So evolution has an inbuilt bias towards simpler phenotypes. And this so I'm not saying this explains all of convergence for sure. I don't think it explains at all why integers converged. Okay, but I think it does hopefully explain some convergences you see at these more molecular levels, and perhaps also at things like shapes like leaves and teeth. It also tells you that what's kind of interesting if you are a philosopher of biology, there's long arguments about ultimate and proximate causes in biology, and this is an example of what I think is an ultimate structuralist cause. It's ultimate because it'll happen again and again. Real type of life, again, start from the same beginning, and you're going to get a very similar RNA structure, for example. I also discovered, I think, that function is apparently very easy to find for RNA. So easy that if you look at the distribution of RNAs that we currently see in nature, they look exactly like the ones I just get from randomly sampling sequences. Now, that's not how they came about, okay? But it tells you that they didn't have to look very far to find function. We're doing the same for proteins, which would be the big question, and Zachary is sitting in the audience somewhere. You can ask him more about it. We think that we found some evidence already that there's definitely some selection there. So that story doesn't hold for proteins, but it's still true that if you look at proteins, our work that we found so far shows that a lot of very similar principles are there. And it's apparently much easier to find function than we had ever thought was possible. And that's super interesting because it tells us that somehow life is easier to find in that hypermolecular space than we would expect it. And so the really big question is then why is it the case that small, tiny fraction of all of the biological possibilities that we keep finding again and again? So why are the things that are easy to find, that this inbuilt Occam's razor allows us to find, why are those enough for life? But why is life set up in such a way that it seems to look in the right spot of life? Or could it be that if I looked somewhere else, life would also been easy? Or is something special about this particular subset that it finds? I don't know the answer to that, but I think it's a definitely question worth pursuing. So with that, I want to thank you for your time and attention.
Please. Oh, thank you so much, Ard, for that incredibly thought-provoking lecture. I'm going to stand over this side so I'm not blocking the view of the camera, because we're going to have some time for Q&A now. We've got a couple of roving microphones, or one roving microphone somewhere. If you have a question, raise your hand and then wait for the microphone to come to you, and so everyone can hear what you're saying. But yeah, we've got about 10 minutes now for questions. And let's start in the front here.
Thank you. With regard to genetic engineering, would you be saying that there are lots of more complicated possibilities for something that could do something more complex, but you need a bit of intelligent design to actualize that?
That's a good question. Actually, I think about synthetic biology. What I'm saying is that there's a vast set of RNA structures, for example, that have never been sampled by biology. You need some kind of human intervention, some intelligent design to get there, because it's hard to find. The interesting question would be, would those never yet existed structures be biologically interesting, or not? I don't know the answer to that question, but I think if I was a synthetic biologist trying to do cellular biology that hasn't been done before, I'd be looking in that space because it's huge. It's unimaginably huge, and it's been completely unexplored. But it's an excellent question, of which I've spent a lot of time thinking, but I don't quite know. It turns out it's also harder than it looks to find those structures because they're also a lot less mutationally robust. So a few mutations and they fall out back towards simple ones again. But it's interesting. Yeah, there might be, a new company you could spin out, right? It's never yet seen structure.
Reflecting on comments by Dennis Noble, how applicable are models constructed in silico to systems that are in vivo and basically in aqueous medium?
Thank you for that question. So I think this always depends, right? So a model is only as good as the assumptions that have been put into it. So for this example I gave you of RNA secondary structure, this does a really good job of predicting what we find in vivo. And it's explained many things. This is why, for example, in the Shostak paper, he takes those things, he runs them through, he takes the sequences that come out, he runs them through a predictor, and this tells him a lot about what he's found. I know Dennis quite well. Obviously, if you're looking at a more complex system, right, you need to make sure that you have all the right ingredients in it for it to work. I might turn this around and say, these computational principles I'm talking about, like this bias towards simplicity, this is something that holds very generally, doesn't really depend very much on the details of the machine that it's on, as long as the machine has certain computational powers. These principles should hold. And so some of these principles may hold independently. I mean, Dennis has, we could do a longer story about some of his worries and complaints about the computational models of life. But for these kind of questions, I think this is pointing you in one particular direction that I think is might be more reliable than you might initially think. It's a hope.
Hello. One question. For the lack of positive selection in RNA, you think that could be related to just the amount of time available to develop those structures, or just simply because it comes to a plateau that's simple to develop, but...
Could you ask that again? Sorry.
For the lack of positive selection in RNA, yes, it's only in our career, I believe you're seeing some evidence of that, I think. Yes, so you think that's just a lack of time to be able to develop those structures that can be more and more selected for, or just a lack of space, really, spatially that's functional to start going towards that structure?
So thank you for that question. So the question is basically, if I understood it right, is the fact that I see a limited number of RNA structures because there hasn't been enough time to explore all that space? And the answer, I don't really know. I suspect that what we're seeing is something slightly different because I only need a really small number of random sequences to find all the if I look at my distributions of junctions or my distributions of loops or distributions of voltages, I need, in this case, for a few 100,000 random sequences, and I find that whole distribution. That's very small in biological terms, which suggests that nature didn't keep looking because it found everything it needed. The argument. So the argument would be, once you've found something you need, then you don't keep looking. But obviously, the longer you look, the more you're going of the structure you're going to find. An interesting thing to remember is that because the probability goes as 2 to the minus the complexity, right? You've got to get 1 bit higher in your complexity, you've got double your search size. So it grows, the amount of new stuff you get, you need to look exponentially long. A different story that I didn't tell today is one about symmetry in protein complexes. So proteins form dimers, trimers, tetramers, all kinds of shapes. And we predict and have shown that the distribution of complexities, distribution of symmetries that you find in nature also follows this 2 to the minus k power law. And we see that in the PDB, so the protein database of structures, and we see that in our models. It maps very nicely. There we do think that as we keep finding more and more proteins and crystallizing them and putting it in the database, there are some rare shapes, so typically no symmetry shapes that are much less likely to appear by random mutations that are going to be found. But for the RNA, it doesn't seem to be the case for reasons I don't understand. The only thing I can think of is that somehow function is so easy to find that nature doesn't keep looking. The other option would be, structure doesn't correlate with function, but that would be kind of really nuts if that were true. Yeah, all structural biologists look at me like in horror that I even mentioned that, but I think that's also true.
Yeah, I think we need to make this last question.
Yeah, so that's true. So as a physicist, you seem to think function is structure. As a biologist, I would disagree, because actually protein structure is incredibly easy to predict, but RNA structures is virtually impossible to predict. RNA is so flexible. So I'm not quite sure what you mean by function and if it's biologically relevant.
Yeah, so so RNA tertiary structure is very hard to predict. RNA secondary structure is very easy to predict. The reason,
but secondary structure becomes irrelevant. The tertiary structure is biologically relevant. So I would follow the comment down here that Noble makes, that actually this is a fundamental disparity between physics and biology, between maths and biology. But we can chat about that later.
Yeah, so I think I disagree. Now the reason why we can't do the same for proteins as we can for RNA, there's lots of theories about why that is. The most likely one is that just the amount of data is too small to train the kind of AI algorithms that have revolutionized protein structure prediction. So that may just be a practical question rather than a biological, but maybe there's a biological reason for it as well. It's still true that a secondary structure highly constrains a protein tertiary structure. And the really important point is that these secondary structures are very heavily under purifying selection. In other words, when that ribozyme has a mutation that changes the structure, purifying selection gets rid of it. So the idea that structure and function are linked comes from the purifying selection arguments. The purifying selection tells you that structure is under some kind of strong evolutionary pressure, and that must come from not from the structure itself, but from the function that it produces, and therefore the structure must be importantly linked to the function, including secondary structure. Now, I think the really interesting question, maybe underlying what you're saying, is this story that I'm telling you still true for tertiary structure? Because one secondary structure can have multiple tertiary structures. So if DeepMind comes up with an AlphaFold that works for RNA structure, and I do the same argument, same kind of analysis I did as I did for the secondary structure, was I ran the pick sequences and look what structures I find. Will I still be able to predict the tertiary structure you find in nature, right? Or is there actually some positive selection that happened at that level? In other words, so we know that there's positive selection that's already there at secondary structure, but it's of course effectively working at a 3D molecule, not at the simpler representation. And the question would be, would we then some deviations from the random model, and from my random null model? And the answer is, I would be very surprised if we did that. But the point would be, if this structure was not important, secondary structure was not important for functioning, it wouldn't be conserved, right? But it is conserved. That's a kind of strange thing. The important point is, because something is conserved doesn't necessarily mean that it also appeared there by natural selection. So, for example, a famous example would be, if an eye was directly created by God, okay, and so we have eyes, it's still true that the mutation that would make me blind would be under positive selection. In other words, my offspring would be less likely to have more offspring, and eventually that mutation would die out. But I can't say much about the origin of a structure from the fact that it's under purifying selection. In other words, but I can tell you that it's got functional value from the fact that it's under purifying selection, and the secondary structures are under purifying selection. So that's the kind of mystery that I'm trying to understand.
Wonderful. Okay. Well, we're going to need to wrap up this section of the evening now. Opportunities maybe to discuss further over drinks in a second. But yeah, first of all, thank you so much, Ard, for all of your insights and for the lecture. Really appreciate it. Okay.